Hello everyone, this is Fahim from Exam Essentials. So today our main objective is going to be to solve this Unit 4 paper from Biology Jan 2025. So without wasting any more time, let's just get started. Right, so the first question shows some processes linking photosynthesis to productivity and distribution of biomass in trees. Okay, biomass in trees, right? So box J, photosynthesis, box K, box L, biomass distribution, leaves, wood, and yeah. Okay, so this first thing here is gonna be gross primary productivity. So what is basically this gross primary productivity? It is the total amount of energy that the plant uptakes due to photosynthesis, or the plant obtains due to photosynthesis. This is the gross primary productivity. What is this net primary productivity, NPP? It is the amount of energy that passes to the next trophic level, taking into account the amount of energy that has been lost due to respiration. Let me repeat again. Net primary productivity is the amount of energy that passes on to the next trophic level, taking into account the amount of energy that has been lost during respiration. Clear? All right, so let's just move on. It says that which row of table identifies the box that represents net primary productivity. Net primary productivity, NPP, the box that represents gross primary productivity, GPP, and the box that represents R, respiration. So basically, as you have saw, we have basically marked this up, right? So J is gonna be JPP, K is gonna be respiration, and L is gonna be NPP. So here the answer is gonna be C. Right. So what what are suitable units for NPP, right? Net primary productivity. So basically, I mean, you have to memorize this stuff, but since this thing is a measure of energy, hence D is gonna be the answer. So kilojoule should be a unit of it, right? And this you basically need to memorize that. Meter to the power minus 2, yard to the power minus 1. Wait, let's just move on. The biomass distributed to the leaves, wood, and roots of trees in this forest was 12,500 arbitrary units. Okay, the biomass was distributed 34% to the leaves and 37%, and the rest to the woods of the trees, calculate how many units of biomass were distributed to the woods of these trees, right? So this is basically nothing, just basic math, right? So assume that the total percent this represents is gonna be 100. From 100 we have 34% that was distributed to leaves, and 37% that were distributed to roots. So if you subtract this 34% and 37% from your initial 100%, you're gonna get 100 minus 34 minus 37, you're gonna get 29%, right? And this 29% represent of the biomass of the that were distributed to the woods of the tree, right? So you know that the total biomass was 12,500, right? So 29 by 100 times 12,500 would gives us the biomass that were distributed towards the woods of the tree. Clear on this? All right, so let's just move on. This is how it looks like. Okay, so which row of table shows what can be found in biomass of leaves and chlorophyll, right? So what basically is this biomass, right? Biomass is the total mass of the living organisms, in short. Biomass is the mass of the living organism, right? So and let me just tell you something. How is this biomass formed, right? So suppose these are the small molecules. Suppose this could be either be amino acid or glucose, right? And when these small molecules join together to form a polypeptide, such as proteins or starch or proteins, this is what we refer to as This is what we refer to as addition of biomass. I mean, these insoluble molecules acts forms the biomass of something. So biomass is nothing other than the total mass of a living organism, right? So which row of table shows what can be found in the biomass of leaves and roots of tree, right? So in the leaves we can get chlorophyll and starch, and in the roots we can get, first thing from the roots that we can get is nitrates and not nitrates, proteins. And let's just take a look at it first of all. Right, so as I said, in the leaves we are gonna get chlorophyll and starch, and in the roots we are gonna get starch and proteins, because the roots take up this nitrate ions from the soil, and this nitrate ion combines with sucrose to form proteins, right? This is the thing. So answer is gonna be C. Let's just move on to the next part. Okay. Basic stuff. So what is meant by the term ecosystem? Basic definition, right? It is the area of the environment where we can find interactions between the biotic and the abiotic factors, or where the organism interacts with each other. Ecosystem is where the organisms interact with each other and their environment, right? Right. So let's just move on to the next question. All right, the time of death. Okay. The time of death of mammal can be estimated in a number of different ways, right? The identification of insects present on the body can be used to estimate how long the mammal has been dead, right? Which method uses the number and species of insects to estimate how which method uses the number and species of insects to estimate how long a mammal has been dead? So basically we're talking about forensics, right? And the process of forensics, in which we use insects to identify the time of death, is what we refer to as forensic entomology. So what basically we're trying to do is, suppose this is the dead body, right? And once this dead body has been exposed, it's going to attract different species of insects such as blowflies and so on, right? So when this blowflies arrives over here, it starts to feed on this dead body, and this blowfly starts to lay their eggs, right? Lay their eggs. The eggs then pupate, the eggs then hatches, and they starts to feed on this dead body carcass, and yeah, they begin to grow, and yeah, they become adults, and this procedure repeats itself, right? So basically we can find the life cycle in which each of these insects are. There are going to be other insects such as I forgot the first, give me a moment. One was this blue flies, and there were these other kinds of flies, right? So yeah, there are these different types of insects. So basically we could find out the stage in their life cycle and identify the time of death. This is what I was trying to mean, and this is what we refer to as forensic entomology. Explain how body temperature explain how body temperature and degree of muscle contraction can be used to determine the time of death. Okay, right. So as the person dies, right? So first of all, we have to understand something, that we are endotherms. We are endotherms. We are endotherms. And what do you mean by this endotherms? We are warm-blooded animals. We are warm-blooded animals. Warm-blooded, right? Warm-blooded. Warm-blooded. And we have a constant body temperature of 37 degrees Celsius. Now the question is, how are we maintaining up this 37 degrees Celsius, right? So inside our body, respiration is taking place, and as respiration is taking place, ATP is produced along with heat, right? And this heat that is formed due to metabolic reactions is what we know as metabolic heat. Metabolic heat. Now once the person dies, the body of the person deprives from oxygen, right? And as a result this procedure of respiration cannot take place, right? And as a result, metabolic heat cannot be generated, and as metabolic heat cannot be generated, the body temperature starts to fall. We can then identify fall in body temperature. We can then identify the fall in body temperature and calculate the time of death, because the body cools down in a relatively predictable manner. So there's this graph, and the body temperature cools down in a predictable manner, right? So we could basically find out when the temperature of the body was 37 degrees Celsius, right? So yeah, this is how we can find out the time of death using the temperature. Right, now there is this another procedure that is what we refer to as muscles, right? Muscle contraction. So I mean, this links up the idea with unit 5 a bit, and in unit 5 we have this idea of ATP, right? So ATP binds with actin mass in head and breaks down the cross bridges, right? And I mean, these are all unit 5 stuff. So I'm not gonna go into details into all of this, but let me just tell you something. You need this ATP to maintain the muscles in a relaxed state. ATP is needed to maintain the muscles in a relaxed state, right? So basically, suppose these are your muscle fibers, and um, they have a certain amount of ATP present in them. The muscle fibers have a certain amount of ATP present in them, right? So as the person dies, this ATP store is gonna drop because ATP won't be produced by metabolic reactions, right? So as I said, when a person dies, this metabolic reaction will stop and they will be producing up this ATP, right? So their ATP levels would start to fall, right? So with some simple calculations, we could identify when this ATP level was filled or full, right? And hence we can find an estimation of the time of death, right? Clear? Because if the muscles are deprived of ATP, then they are gonna go to a contracted state, and that state is what we refer to as rigor mortis. Rigor mortis, right? As I said, so basically, for unit 4, you just have to know the fact that energy is needed, ATP is needed to maintain the muscle in a relaxed state, right? And if it doesn't receive of the ATP, then the muscles are gonna go into a contracted state, and that is what we refer to as rigor mortis. I mean, rigor mortis starts generally around 2 to 4 hours of death and can last up to 36 to 48 hours. So yeah, this is what I have written down. So after death, metabolic reactions inside the body stops and metabolic heat is not generated due to respiration. Hence, heat energy gets transferred to the surrounding via the process of convection, conduction, and radiation. Time taken for the body to cool down can be determined, and hence the time of death. ATP is needed to maintain the muscles in a relaxed state. After all of the ATP has been used up, the muscle contracts, and this process is what we refer to as rigor mortis. With simple calculations, it is possible to determine the initial number of ATP and hence finding the time of death, right? So yeah, basic question. Let's just move on. The photograph shows a Canadian lynx and hare. So this is the lynx and this is the hare. From this photograph, I can understand that this is gonna be the predator and this is gonna be the prey. The hare is gonna be the prey, right? So let's just move on. The lynx is a type of cat, and as I said, is a predator of the hare, and the hare is the prey. Graph 1 shows changes in the number of lynx from 1845 to 1895. In 1863, there were 15,000 hares. 15,000. Calculate how many times more the number of hares were than the number of lynx. So let's just find out the number of lynx in 1863. So this is 1863, right? 1860, 61, 62, 63, right? And so corresponding part of the graph, the number of hares is gonna be 20. Do not forget, you have to add into 10 to the power 3, right? So yeah, you have to divide up the number of hairs divided by 20 into 10 to the power 3, 20 into 10 to the power 3. So you'll be getting 7.5. So the number of hairs is greater, right? By how many times? By 7.5 times. Basic maths. I mean, in biology, you do not need to have an understanding of in-depth knowledge of maths, but you have to have an idea of how to calculate this basic maths, right? So yeah, let's just move on. Graph 2 shows a part of graph 1. Okay, sketch a line on graph 2 to show the likely changes in the number of hairs from 1855 to 1888, right? So the graph should go something like this, the number of hairs, because this graph is given inside your book, right? And you have to know something, and that is the number of predators and the number of preys oscillates between two extreme points. See, what do I mean by this extreme point, right? See, as the number of hairs decreases, the as the number of links decreases, the number of hairs are gonna increase. Why? Because these links predate on this, or the links eats these hairs, right? So when the links eats these hairs, their number is going to get decreased, and right, and when the links does not eats the hairs, the number of hairs are going to increase because they are going to reproduce, right? So they are going to reproduce, right? And the number of hairs, um, the number of hairs dying is less than the number of hairs being reproduced, and hence the number of the number of hairs increases, right? So yeah, and again say this, as the number of links as the number of links is the greatest, the number of hair starts to fall. So basically you have to know this thing, right? So they oscillates between two extreme points, the number of predators and the number of prey. Explain the change in the number of links and hairs, right? So basically from here we can see that uh, the number of links decreases as the number, so we have to explain, right? And what do you mean by explain, right? It means that you have to justify, you have to provide an explanation, right? So we can say that the number of links decreases, as the number of hairs decreases. And why is that? This is because these links eats up these hairs, hairs, right? So when the number of links are going to decrease, the numbers of hairs are going to decrease as well, right? See, the number of links is decreasing, the number of hairs is decreasing as well, because these links eats up these hairs, right? And number of hairs decreases uh, as they are hunted by links. And see, the number of hairs are decreasing. Why? Because they are being hunted by the links. Basically, yeah, this is more or less that you have to know from this. See what I've written, number of links decreases as number of hair decreases because links eats on hairs. Decrease in number of links is due to starvation. Number of hairs decreases as they are more hunted by links, right? So in many ecosystems, the relationship between one predator species and other prey species are less predictable. So just two reasons why the relationships are not always predictable, right? So basically, we have just got a basic understanding from here that as number of predators increase and decreases, the number of prey also increase and decreases, right? And one reason could be due to predation, increased predation and so on, right? But what are some of the other reasons? Some of the other reason could be due to a disease that would lead to the population of prey to decrease, right? One predator species or prey to decrease, right? And um, there may be also lack of food that the prey eats, right? Which would cause their number to decrease, right? Because you have seen here, right? As the number of links decreases, the number of as the number of hairs decreases, the number of links decreases as well, right? So basically, as the amount of food decreases, the amount of food for the predator decreases, the number of predator decreases as well, because the predators are going to need something to give them energy and to reproduce, right? So yeah, as the thing that their predator is decreases, their number is initially going to decrease as well, right? and there could be some sort of a disaster, ecological disaster, or there could be some sort of a disease that would cause the numbers of predators or prey to fall, right? This is I have written over here. Right, let's just move on to the next question now. All right, so the diagram shows the structure of chloroplast and is related to its role in photosynthesis, right? So this is a basic structure of chloroplast, and you basically need to have a basic understanding of all of this, right? So yeah, so on the thylakoid membrane, the light dependent stage of photosynthesis takes place, and inside the stroma, the light independent reaction takes place, right? And what does hydrogen ion accumulates for the use in photosynthesis? So during the photolysis of water, so what is photolysis, right? So as sunlight falls on water, it breaks down to form H plus, right? H plus, right? So the hydrogen ions are going to accumulate in the inside of the thylakoid membrane, right? So it accumulates over here inside of the thylakoid membrane, right? Right, so yeah, this was more or less about it, and you should have an idea of how to label all of this, because this is a repeated question and it generally shows up, right? In sometimes they may ask you to label all of this, and sometimes they may give multiple choice question based on its function, right? So row of, which row of table shows why light independent reaction takes place and light dependent, right? So light dependent on the thylakoid membrane, that is going to be T, and light independent is going to be in the stroma, that is going to be R. So T and R. So see, T and R. Why do hydrogen ion accumulates, as I said, inside the thylakoid? So that's going to be S, right? So the diagram is 16,000 times larger than an actual chloroplast. Okay, the diagram is 16,000 times larger than an actual chloroplast. Calculate the surface area of the chloroplast using the formula uh, using the formula 4 pi r square, right? So the diagram is 16,000 times larger, right? So basically, I'm sorry, but there was a line here, there was a straight line over here. So basically, using your scale, you have to find out this diameter. First of all, you have to find out this diameter. Using your scale, you have to find out this diameter of the chloroplast, and then divide the diameter by 2 to find out the radius. Then you plug this all of this into the formula 4 pi r square, and you divide by 10 to the power minus 6 to add up this prefix of u, and it says that the diagram is 16,000 times larger, right? So you have to divide by 16,000, and hence you're gonna get, the radius, right? Radius in, you're gonna get the radius, right? And then what you're gonna do is you're gonna put the radius here, and you're gonna get 130 micrometer square. Here so far? Yeah, so what I did was I found the radius using a scale. And I divide by 2. I mean, it basically is the same as 10 to the power minus 2, right? And then I converted it into micro. So yeah, micro, and it said that the diagram is 16,000 times larger, so I divided it by 16,000, found the radius to be 3, and then I inserted it in this formula 4 pi r square, and yeah, that is how I received out the actual surface area. And explain the advantage of thylakoids in a chloroplast of having a large surface area, right? So as I said, the light dependent reaction of photosynthesis takes place on the thylakoid membrane, right? And inside the thylakoid membrane we have these different structures known as photosystem 2, photosystem 1, electron transport chain, ATP synthase, and so on, right? So having a large surface area means that more of these structures could be packed inside the thylakoid membrane, and as more of this could be packed inside the thylakoid membrane, more ATP and reduced NADPH could be formed, and reduced NADP could be formed, and this is then fed into the Kelvin cycle, or the light independent reaction inside the that takes place inside the stroma. And as the amount of this increases, the rate of light independent reactions increases as well. So yeah, this is what I have written over here. Can be packed with more ATP synthesis photosystem electron transport chain. This will lead to faster production of NADP and ATP in the light dependent reaction that can be used in the Kelvin cycle or light independent reaction, and light independent reaction takes place inside the stroma. So yeah, and lead to an increase in the biomass. Clear? All right, let's just move on. Okay, so in the light dependent reaction, ATP and reduced NADP are generated, right? So these products are used in light independent reaction. The diagram shows some of the light independent reaction. Okay, the diagram which describes ATP is produced in the light dependency, right? So ATP is produced due to the phosphorylation of ADP. By phosphorylation of ATP, ADP, adenosine diphosphate, requiring energy, right? Let's just move on. When one molecule of glucose is made in the light dependent reaction, the ratio of ATP to reduce NADP is 1.5:1. You have to show this ratio using the diagram, right? So yeah, basically uh, basically just ignore up my text for a while. Let me just explain, right? So this is RUBP, right? So let's just calculate for one, right? Let's just start with one. Let's just start with one Kelvin cycle, right? So let's just, instead of 3, we'll be starting with one RUBP, right? So RUBP is a 5 carbon compound that combines with the carbon from the atmosphere to form a 6 carbon compound, right? And the 6 carbon compound is then converted into glycerate 3 phosphate. Glycerate 3 phosphate, right? Glycerate 3 phosphate. Glycerate 3 phosphate is a 3 carbon compound. 3 carbon compound. So your glycerate 3 phosphate is a 3 carbon compound. So first of all you have this RUBP that combines with carbon dioxide from, and this is a 5 carbon compound, and that this combines with one carbon from the carbon dioxide in the air via a process known as carbon fixation to form a 6 carbon compound. 1 2 3 4 5, right? And this 6 carbon compound is highly unstable and breaks down to form 2 3 carbon compounds, and this 2 3 carbon compound is what we know as GALP, GALP, glycerate aldehyde 3 phosphate, right? And the GALP is is GP, glycerate 3 phosphate, and the GP is then converted into GALP, and it requires a ATP and reduced NADP to convert one ATP and one reduced NADP is used to convert this GP into GALP, right? So as there are two GPs, so we would basically need 2 ATP and 2 reduced NADP, right? So then we have this 3 carbon compound curve, then one of the carbon goes on to produce glucose, right? And the other 5 carbon joins together to form to regenerate that RUBP, right? And when the other 5 carbon compound combines, they're gonna need 1 ATP. This was for 1 Kelvin cycle, right? But in glucose you have 6 carbon, which means that the Kelvin cycle needs to run 6 times, right? And if the Kelvin cycle needs to run 6 times, so basically you are having 2 ATP here and 2 NADP here, right? So this would be multiplied by 6. So that is going to be 12 ATP and 12 NADPH, right? And 12 ATP again, right? So the total amount of ATP was 12 plus 12. Oh, sorry. This is gonna be, this was 1, right? So as the cycle is gonna run 6 times, so this is gonna be 6 ATP, right? So you had 12 ATP here and 6 ATP here, so total of 18 ATP. 18 ATP. And reduce NADP, right? So you had 2, right? You had 12 NADPH is to 12 NADPH. You then simplify this. See, 18 is to 12, and you then simplify this, and this is what it comes, that is 1.5 is to 1. Clear? All right, so let's just move on. Let me use all of this because it is looking pathetic, disgusting. Right. If you are facing any problems in any of these questions, or in fact this biology syllabus, please let me know. I mean, I would be more than glad to help you all, because I have faced the same problems, right? And there wasn't anyone there for me to help me and to support me, right? So I'd be more than glad to help you all. And honestly, this is what I have to do in my university as well, right? So yeah. Explain why a small proportion of GALP produced is used to make glucose. All right, so the first thing is that only one, let me just draw this up again in short. Only one from this GALP is used to make glucose. The other four are joined together to make RUBP, right? And the RUBP is needed to run up the what? To run up the Kelvin cycle, or to run up the process of carbon fixation, right? Because using this RUBP, this carbon from carbon dioxide combines to make this 6 carbon compound that is highly unstable, right? So if you didn't have RUBP, so your carbon fixation process wouldn't run as well, right? So hence you need this. Hence a small proportion of GALP is used to produce glucose. So basically in each cycle, one of the carbon from 2 GALP is used to make glucose. This is because GALP is converted into RUBP that could be used to run the cycle of Kelvin cycle repeatedly. Otherwise carbon fixation would not take place, and hence light dependent reaction will not occur, right? All right. So moving on to the next question. Right. Peat bogs are waterlogged areas with low pH. Okay, peat bogs are. So basically, what is this peat bog, right? So when you search up this image, it shows that these surfaces of water, surfaces like this. There is this hole, and this is filled up with water, right? So these peat bogs are waterlogged and they have a low pH because the atmospheric pH cannot dissolve into the water, and the remaining oxygen is used up by bacteria or aerobic organisms. So the condition is anaerobic over there, and the pH is obviously going to be low. Pollen grains in peat bogs can be used to determine the types of plant that grew in a particular area in the past. Okay, the diagram shows the types of pollen shaded found in a site in New Mexico. Okay, cereal crops and beans are sources of food for humans and their animals. Okay. Explain the abundance of these 3 types of pollen grains as shown in this diagram. Okay, so from here I can see that the abundance of cereal crops initially in the past were smaller, but recently they increased, right? The recent increase could be due to cultivation for supporting food, for to feed the animals and the humans, right? So explain. Whenever there's an explain question, you cannot just say that in the past they were smaller and recently they increased. You have to state why. You have to justify. So why they increased? Due to increased cultivation. For beans, this is going to be the same. In the past it was smaller, but recently it increased due to cultivation. And for trees in the past were abundant, trees recently start the population, the number of trees starts to decline presently. Why is this? Because the population is increasing and people is basically cutting down trees and forests to get woods to build up houses or to to keep themselves warm by using by burning them, right? So I've written that here. So abundance for cereal crops increased more recently due to increased cultivation, whereas abundance for trees was greater in the past and then recent recent as trees are cut for wood to build homes. Increase in abundance in beans is due to increased cultivation of the beans for food. Right. So let's just move on. In 1950, the preserved body of a man was found in a peat bog in Denmark. The man died over 2000 years ago. The graph shows maximum and minimum temperature and the mean rainfall in Denmark. So, max temperature, mean temperature, and rainfall. Let me just mark this up. This is the max temperature, mean temperature, and this is the mean rainfall. Okay. What is the difference between the maximum and minimum temperatures in May? So in May, in May, May, this is the maximum temperature. So it comes to be 16, and this is the minimum temperature, and it comes to be 8. So 16 minus 8 is gonna be 8. That should be the answer. Right, so explain how the conditions inside the peat box preserved the body of this man from decomposition, right? So, in the first part of the question, we saw that peat bogs are waterlogged and they have low pH, right? So, who carries out decompositions? Microorganisms does, right? Microorganisms, or these are decomposers, right? And decomposers are living organisms, and if something is living, they possess up enzymes, right? They possess up enzymes. So, the enzymes are sensitive to temperature and pH, right? So low pH means that the enzyme couldn't efficiently decompose that because their active site were denatured and so on, right? So yeah, one thing could be that, and another thing would be, see, the highest temperature, the maximum or the highest temperature is over here. See, the highest temperature, if I'm not wrong, this should be the temperature, right? And the highest temperature is below 24 degrees Celsius, which means that the rate of decomposition is slowed as well. The highest temperature is below 24 degrees Celsius, right? Which means that the rate of decomposition is slowed as well, because enzymes work fast at 37 degree Celsius. And another thing is that rainfall, right? Rainfall. So as you saw that peat bogs were waterlogged, right? So generally in the amount of rainfall in Denmark was high as well, except for 1 month. Except for let me just see the month. Except for this February. Except for this February, right? So that meant that the pit box were always filled with water and the conditions were anaerobic, right? So yeah, that prevent decomposition as well. So inside the peat box, low pH inhibited the process of decompositions by the decomposers as their enzymes could be denatured. Mean temperature in Denmark is below 24 degrees Celsius, which slowed down the enzyme activity of the decomposers and prevent decomposition. Peat box covered the body and reduced body exposure, which protected the body from insects such as blowflies, beetles, scavengers, and so on, right? The first part of the question. I mean, we did a question similar to that, right? So, as the body were exposed, they attracted blowflies and so. So, as peat box covered the body, these insects could not be attracted towards the body. And mean rainfall in Denmark is generally high except for the month of February, which means that the peat bog remained filled with water and the conditions were anaerobic, right? So yeah, let's just move on to the next question.
Let me guess. You are watching videos, taking notes, maybe even grinding late at night. But the marks, they are just not going up. You are stuck, and it's frustrating. But here is the truth, it's not your fault, it's the method. And that's exactly where Exam Essentials makes the difference. We at Exam Essentials actually offer two very powerful courses to navigate you towards success. The first one is the most popular high impact crash course. If you are just 4 to 6 months away from your exam, this course is going to be a lifesaver for you. The high impact crash course for the October 2025 and the January 2026 is already available. And the second course that we at Exam Essentials actually offer is the complete 1 year course. This is basically for the students who want full support from day one to exam day. And the best part, our students' results speak for themselves. Both the courses are actually open ahead of every exam session. No matter whether you are going to give your exam for the May 26, Jan 27, May 25, or October 25, doesn't matter. We always offer these two courses before every upcoming exam session. To join or know more, just check the description box below. You will find our WhatsApp number and Facebook page link right there. Let's turn your effort into results together.
So moving on to the next question, that is question number 6. All right, so it says that the development of antibiotic resistance is a global concern. Okay, a new type of drug is developed against bacteria. Okay, the drug has a number of effects on bacteria, including making bacteria more susceptible to antibiotics. Okay, so a drug has been developed, and the purpose of the drug is to make the bacteria more susceptible or vulnerable to the use of antibiotics. Okay, the drug is itself not an antibiotic. Okay, the drug is not an antibiotic. Okay, so state the meaning of the term antibiotic. Antibiotic is a medicine that changes the structure of the cell wall of the bacteria, causing the bacteria to die, causing the bacteria to eventually die. So how can what the antibiotic does is, bacterias, assume that bacterias have a cell wall, right? So antibiotics targets of the cell wall, right, and the damage of the cell walls. And once the cell walls are damaged, water and ions can move into the cell by osmosis and cause the cell to burst and cause the lysis of the cells, basically, right? So let's just check up the questions. So the first question is, okay, term antibiotic. So it is a medicine that affects the metabolic pathway of pathogen and even directly kill it by destroying the cell wall, by destroying the structures like cell wall. Okay, so when it affects the metabolic pathway, the antibiotic is what we refer to as bacteriostatic, and when it damages structures like cell wall, the antibiotic is known as bactericidal. Bactericidal. Okay, let's just move on to the next question. In 2019, 1.27 million deaths were due to antibiotic resistance. It is estimated that this will rise to 10 million per year. This will rise to 10 million per year. What is the percentage increase in antibiotic? What is the percentage increase in the number of deaths due to antibiotic resistance, right? So this is the final, this is the initial. So final minus initial divided by this initial times 100 would give us the percentage increase, right? So yeah, basic maths. This is what I have done. Final, this was final, and this was initial divided by initial number times 100. So this has given us a percentage increase in the number of deaths due to antibiotic resistance. Okay, explain why it is important that these new types of drugs are developed. Okay, right? So why new drugs have to be developed? Because these bacteria could be resistant to these existing drugs, right? So you have to develop these drugs to destroy up the pathogens that are resistant, and this would ensure that less number of people dies from infection that are caused by these lethal bacteria. So to destroy the antibiotic resistance so that fewer people dies due to bacterial infection. Right, so let's just move on to the next question. The diagram shows the structure of the cell wall of a bacterial cell. Okay, it shows the cell membrane. The molecule in the outer membrane varies on different types of the bacteria. Okay, varies due to different types of the bacteria. This is the cell wall structure. We have a outer membrane here. So when bacteria were treated with new drugs together with antibiotics, the antibiotics were effective at lower doses. Remember, it said that when you use this drug along with the antibiotic, it increases the effectivity of the antibiotic, right? It makes the bacteria more susceptible to the antibiotic, like related to the first part of the question. The new drug attached to the outer membrane, okay, suggest why new drug resulted in, okay, right. So as soon as I hear the term membrane, what is the function of this membrane, right? So what the cell membrane does, it basically controls the movement of the ions into and out of the cells, right? It affects the permeability of the cells. Whenever you hear the term membrane, the first thing that should come into your mind is about permeability, right? So as the bacteria, as this drug attaches itself to the outer membrane, it would affect the permeability of the membrane, right? It would affect the permeability of the membrane and hence the movement of substance into and out of the cell, and as a result the antibiotic could efficiently destroy it. So yeah, I have said that drug changes the permeability of the outer membrane, hence lower doses of the drug is needed to disrupt the peptidoglycan layer of the bacterial cell. Okay, let's just move on. The graph shows the effects of three antibiotics on the species richness of gut flora. So what basically is this flora? Flora is the total number of microorganisms of different species that could be found inside your body, right? So describe two conclusions that could be made about the effects of these three antibiotics on the species richness, right? So basically all of these three antibiotics, as you can see, so basically all of these three antibiotics causes the species richness to decrease, right? All of these antibiotics causes the richness of the species to decrease. However, the decrease is greatest with antibiotic C and is the least with antibiotic A. So these are the two conclusions that we could deduce from this graph. Antibiotic C causes the greatest decrease in species of the gut flora, and all the antibiotics causes the species richness to decrease, right? These two. You can also add up a follow-up point, that is antibiotic A causes the least decrease in species richness of the gut flora. Right, let's just move on. Explain the advantage of adding this new drug when treating a bacterial infection with antibiotic C. Okay, so what this question says is about, as we know from the earlier part of the question, when you're using up this drug, it increases the susceptibility of the bacteria to the antibiotic, right? So as we can see, species antibiotic C causes the greatest decrease in species richness, right? So if we use the drug with antibiotic C, lower doses of C would be needed, right? Lower doses of C would be needed to destroy the bacteria. And if lower doses of antibiotic C is needed, it would ensure that not much of the species richness of gut flora is damaged. Like the gut flora isn't damaged that much, right? Because gut flora competes with pathogens or any other type of microorganisms that are lethal for nutrients, right? And when they does this competition, it prevents these pathogen microorganisms from growing and causing diseases, right? So yeah, this is what I have basically written down in my answer. So let's just take a look at it. Lower concentration of antibiotic C will be needed to kill the bacteria. Right? This will prevent the destruction of the gut flora, which would compete with harmful bacteria for resources. As lower concentration of antibiotic will be required, this will prevent any adverse side effect as well. Because whenever we are using up the drug, we need a lower concentration of the antibiotic for making the bacteria susceptible to that antibiotic, right? So basically, using a lower concentration would prevent any adverse side effects like vomiting, nausea, and so on, dizziness, right? Loss of appetite, and so on. So let's just move on to question number 7. Right, so carbon dioxide, methane, and nitrous oxide are greenhouse gases. Nitrous oxide is produced by bacteria in the soil. Nitrous oxide is produced by bacteria in the soil, right? So step 1, anthropogenic source of carbon dioxide and 1 anthropogenic source of methane. What do you mean by anthropogenic, right? Anthropogenic means something that is caused due to human activities, right? So carbon dioxide it is gonna be combustion of fossil fuels, and for methane is gonna be cattle farming, right? So yeah, this is what I have written down in my answer. Describe how greenhouse gases causes global warming, right? So what the greenhouse gases does is that, suppose this is the surface of the earth, right? And this is the rays of the sun. So short red infrared radiation or short wavelength of light can penetrate through the surface of the earth, and once it is re-radiated back, the greenhouse gases, suppose these are the greenhouse gases absorb this radiation, right, and prevents it from escaping to the outer atmosphere, right, and the gases traps up the heat, and as a result it causes the surface temperature to rise. Clear? As the gases traps up the heat that has been re-radiated from the surface of the earth, it causes the temperature of the surface of the earth to increase, right? Because the gases cannot escape the atmosphere. This is what I have written. Greenhouse gases traps longer wavelength of light that has been re-emitted from the surface of the earth. This prevents the light from leaving the atmosphere of the earth and causes the surface temperature to rise. This then results in global warming. Greenhouse gases accumulate on the upper atmosphere. Right, so let's just move on to the next question. The graph shows the changes in mean global temperature from 1975 to 2023. 1975 to 2023. Predict the change in mean global temperature in 2050 from the data in this graph, right? So basically you just have to make a prediction, right? So first thing is that from here I could see that the highest number of boxes is 4, and from here I could see that the lowest number of boxes is 3, right? So basically I have drawn that 3 and 4. So I have drawn that in a similar manner, and by 2020 it should be around 1.10, so yeah, it is basically a prediction, and many of the students got this question wrong in their exam. So basically, if you did get it wrong, you shouldn't worry that much, because it's basically a 1 mark question, provided that you did others correct, right? So the greenhouse gases have different warming potentials, okay, over over 100-year time scale. 1 ton of methane would generate 28 times warming potential of 1 ton of carbon dioxide, right? So methane is gonna be 28 times into carbon dioxide, and 1 ton of nitrous oxide NOx would generate 265 times of the warming potential of 265 times of the warming potential of carbon dioxide. Calculate the ratio of warming potential of nitrous oxide to methane, right? Nitrous oxide to methane. So this is going to be 265 times CO2 dot 28 times CO2, right? So this thing, the ratio means the division, right? So you can cancel out carbon dioxide, right, and hence simplify this 265 and 28, right? So this is what I have done in my answer. So let's just take a look at it. So yeah, see, and I get a ratio of 9.5:1. Okay, let's just move on to the next question. Carbon dioxide can last in the atmosphere for thousands of years. Methane can last for about 12 years. Nitrous oxide can last about 121 years. Discuss the implications of the data and warming potential when scientists are deciding how to tackle climate change, right? So first of all, they should be considering to decrease the amount of nitrous oxide, because that is the most potent greenhouse gas. See, as I said, it has the highest warming potential. And then they should tackle up the carbon dioxide level, because it has long lives, right? Because they can last in the atmosphere for thousands of years. And nitrous oxide needs to be tackled first, and then carbon dioxide, and then methane. However, methane is also a potent greenhouse gas than carbon dioxide, which means that it is a stronger greenhouse gas than carbon dioxide. But first of all, we have to tackle up this nitrous oxide, then carbon dioxide, and then methane, right? So let's just move on. Methanotropes are a group of bacteria that can absorb methane and use it as a source of energy, and as a material to convert it into biomass. So basically they can convert it into biomass, okay? Most methanotropes grow best in methane concentration of 5,000 arbitrary units to 10,000 arbitrary units, okay? This is greater than methane concentration in the atmosphere, all right? Scientists wanted to identify the methanotropes that could grow at lower methane concentration, as they would help to reduce the levels of methane in the atmosphere. Alright, scientists wanted to identify methanotropes that could grow at lower methane concentration, as they would help to reduce the methane levels in the atmosphere. Okay, the graph shows the growth curve for two types of methanotropes grown in a methane concentration of 500 arbitrary units, right? The increase in turbidity of cell culture was to assess the growth of the methanotropes, okay? Turbidity measurement for methanotrope A and methanotrope B. Explain how this graph can be used to compare the growth rate, right? So basically we are using the method of turbidity, right? So what we can say is that we can draw the line of base fit first of all for each of these, and then find the gradient, as the gradient would give us the growth rate, right? So yeah, so higher the value of turbidity, the greater will be the growth, as the increased amount of bacterial cell would cause less light to be transmitted, right? I believe you have an basic idea about turbidity by now, colorimetry. So as the rays of light passes through this solution, and if the solution is kind of deep or thicker, less of this light would be transmitted through this solution, right? And the light is then detected by a detector, and this gives us the value of turbidity. It's basically colorimetry colorimetry experiment from AS chemistry, right? So yeah, let's just move on and use the line of base fit and find the gradient to measure the growth rate of each type of bacteria. All right. Suggest why low methane concentration are not able to support the growth of some methanotropes, such as methanotrope A, because as we have saw that they would require up this methanotrope to give me a moment, uh, to convert it into their biomass, right? So they would need methanotrope to do something, right? For instance, respiration, right? So I have said that less respiration in bacteria takes place, and as a result they cannot make enough ATP to support growth. So just why scientists analyze the cultures of methanotropes B for the production of nitrous oxide and carbon dioxide, right? So methanotropes B had the highest growth rate, right? And as they grow, they're gonna use up, they're gonna make amino acids for growth and proteins, right? Amino acid for growth and proteins. So the base unit of making this amino acid is gonna be nitrate. So there's a possible risk of nitrous oxide forming from here, and as they're growing fast, it means that they are also carrying out respiration at a greater rate, right? Respiration at a greater rate, and once they carry out respiration at a greater rate, they are going to produce up carbon dioxide, right? that's gonna increase the levels of atmospheric carbon dioxide as well, right? So we are more interested about the adverse effects that this methanotrops B could have, right? So yeah, that's what I have written. They had the highest growth. This means that they respire more and produce more carbon dioxide. So to provide them with energy for growth. And due to increased growth, nitrous oxide waste products may be greater as well due to increased protein synthesis. Nitrous oxide and carbon dioxide may cause more climate change than the reduction in methane could. Right? Because we have saw that carbon dioxide has a prolonged effect and nitrous oxide has the highest warming potential. Alright, so let's just move on. Since the early 2020s, the number of cholera cases and deaths due to disease are high as world is facing its seventh pandemic of cholera. Alright, cholera is caused by ingestion of food or water contaminated with bacterium Vibrio cholerae. Ingestion means something that you eat, right? The diagram shows the distribution of cholera cases by age group and sex at Malawi in the first 9 months of 2022. Okay, first 9 months of 2022. From the graph here, I could say that cholera is cholera affects more um, males than females. First observation, and highest amount of cholera cases is for the age group of 21 to 30 for both males and females. Excuse me, sorry for that. So these were two of the observations that I could make from the graph. So, oh, all right. Make up conclusions, right? So this is what I've basically said at this point. Number of cholera cases is the highest in age group of 21 to 30 for both males and females, and males generally have higher cases of cholera than females, right? Let's just move on. Vibrio cholerae produce a toxin made up of several subunits. Okay, genetically modified rice plants are grown that produce rice grains that contains one of these subunits. These subunits can be used as a vaccine. Okay. The grains were harvested and ground into a powder that could be eaten. In a clinical trial, three groups of people were given one of three doses of this rice powder vaccine. Another group was given a placebo. Okay. The changes in the levels of antibiotic against subunit in their blood was measured over an 8-week period, right? So basically a vaccination type of question. And you can, I mean, basically they are using up a rice plant, right? Genetically modified rice plant, and the rice plant contains one of the toxin that the bacteria, that the cholera bacteria carries, right? So I mean, this is what I mean, basic vaccination type of question. So yeah, keys for rice powder and for placebos, right? For rice powder, I could see that it is going to be greater than the placebos. And also one thing for sure is that the error bars, there are error bars, right? All right. For 3 gram, same, rice powder has a greater effect in the levels of antibody than placebos. For 6 gram, rice powder again has the greatest levels. Then, and what I could see from here is that these error bars aren't overlapping, which means that the datas are significant, right? There is significant difference in the datas. Right, so let's just move on to the question. Describe suitable placebos for the use in this clinical trial, right? So a suitable placebo would be rice grain without the subunit, normal rice powder that does not have the subunit, and concentration. Oh, another thing is that when you are using a placebo, you have to make sure that the concentration remains the same as the antibiotic being tested, or something, or a medicine that is being tested, right? So normal rice powder that does not have subunits for of the toxin, and concentration of this needs to be the same as the rice powder vaccine, right? So the scientists running up the clinical trials concluded that the doses of 6 gram of rice powder vaccine was needed to stimulate an immune response, right? Because yeah, you can see that changes in levels of antibodies is the highest, and also the error bars doesn't overlap, which means that the data are significant for 6 gram. However, for 3 grams and 1 gram, the error bars are overlapping, right? Okay, so state why they came to this conclusion, because the fewer error bars were overlapping, right? Which means that the data were significant. Um, okay. Scientists found that the antibodies binds to different toxins produced by another type of bacteria. Okay, explain why these antibodies produced as a result of the rice powder vaccine could bind to different toxins. So antibodies binds to antigens, right? Suppose this is the antibody and this is the antigen, right? So antibodies is going to do is what they are going to bind to this antigen, right? They are going to bind to this antigen, right? So that type of bacteria would have similar antigen, and also there's a scope that the rice powder was contaminated with other type of bacteria that produce the toxin, right? So this is what I've written. Toxin produced by other type of bacteria has similar antigen, and as a result the antibodies could bind to different toxins. And also there is a possibility that the rice powder was contaminated with other type of bacteria that produce the toxin, right? That produce the toxins. Let's just move on to the next question. Some people in clinical trial did not respond to rice powder vaccine. Okay, the scientists determined the proportion of different type of bacteria A, B, C, D and others in the gut flora of these non-responders with those of the responders. Okay, the charts. Compare and contrast the results for responder versus non-responder, right? So I could see that for non-responder, this bacteria A was the highest. This bacteria was the highest, and for and highest than these responders. And also what I could see that bacteria BCD, bacteria BC, like this bacteria BCD along with this other type of bacteria is lower for non-responder than for responders, right? And compare and contrast, and yeah, more or less this is gonna be it. And let's just see if I could find anything, because I have written down 3 points over here. I'm trying to find out a fourth point if applicable. um you can say that uh this is bacteria D, if I'm not wrong. Bacteria D is the lowest in non-responders, right? Bacteria D is the lowest in non-responders. So yeah, non-responders have higher proportion of bacteria A than responders. Non-responders have lower proportion of other bacteria than responders. Responders have higher proportions of bacteria B, C, D than non-responders. And both responders and non-responders have highest amount of bacteria A, right? So let's just move on. Give two limitations when using the data to determine if the type of gut flora could cause a person to be a responder or non-responder. Okay. So the first thing is that we basically do not know the gender of that person or the age of the person, because the gender of the person could vary and hence the bacteria. And also we do not know that the person and also we do not know that either the person has some pre-existing sort of disease or not, like stomach ulcers or anything, right? So yeah, gut flora could vary from person to person on age and gender. Either the person has any other diseases or not is not known, and cannot identify which bacteria is the cause of this, right? So let's just move on. Last question of this paper. The human genome contains sequences of DNA called endogenous retrovirus ERVs. Okay. The ERVs result from infection with retroviruses. So what are these retroviruses? They have the enzyme reverse transcriptase, and the function of this enzyme is to convert RNA to DNA. Right. Retroviruses are a group of RNA virus that have replication cycles similar to that of HIV, human immunodeficiency virus. Okay. and the replication cycle is, and the replication in latency, right? They follow the lysogenic cycle, which means that as the host cell host cells DNA replicates, the bacterial DNA replicates as well. But this bacterial DNA or the viral DNA produces a repressor protein, and the function of this repressor protein is that it hides up the bacterial DNA from being transcripted, which means that mRNA is not formed, right? And as a result, tRNA and transcription does not result in the production of viral proteins, right? And hence viral cells are not produced as well. So let's just move on to the question. Some of the ERBs in human genomes have been present for millions of years because ERBs are passed through many generations. Explain how retroviruses introduce ERBs into human cells and how ancient ERBs can be transmitted to offspring in families, right? So basically, this is gonna be the answer that could be similar to HIV cycle, right? So first of all, it would attach to the host cell, and then it would release up the viral DNA into the cytoplasm of the host cell, and the reverse transcriptase would convert this viral RNA into viral DNA, and the viral DNA would then get inserted into the host cell DNA due to the enzyme integrase, and yeah, they are gonna be replicated in a lysogenic pathway and hence will be transmitted over generations. So this is what I have written over here. Retrovirus with its antigens attaches with the host cell in a human. The virus then releases its RNA into the cytoplasm of the host cell, which gets converted into viral DNA by the enzyme reverse transcriptase. The enzyme integrates, then incorporates this DNA into the host DNA. Each time the host DNA replicates, the viral DNA replicates as well using lysogenic pathway. The viral DNA produces repressor proteins that prevents transcription and translation of viral DNA, that prevents transcription and translation of viral proteins. Viral DNA as a result become parts of the host genome, or as the part of the genome of the individual, and is passed over to many generations, or passed over to future generation via reproduction. What do you mean by genome? Genome is sum of all of the genes of someone, right? Of an animal. We have two more questions to go. One group of ERVs code for viral proteins. In normal cells, these proteins are not synthesized due to epigenetic regulation, right? Explain how epigenetic, I mean basic A is stuff. So it could either be due to histone modification or methylation, right? So it prevents that viral, it prevents the code that it prevents the DNA that codes for viral environment protein from being expressed, and hence transcription and translation is prevented. Epigenetic modification prevents the. Give me a moment, guys. Sorry for that. Epigenetic modification prevents the expression of viral DNA that codes for viral protein. This could either be because of histone modification or DNA methylation. As a result, mRNA for viral protein is not produced by transcription, and hence translation does not produce the viral envelope proteins. Basic is stuffs. In cancer cells, the viral envelope proteins are synthesized and incorporated into cancer cell membranes. Okay. Explain why viral envelope proteins could be used as vaccine against cancer cells, right? Vaccines against cancer cells. So you have to justify, right? Suppose this is the cancer cell and this has this antigen on its surface, right? Viral and envelope proteins, right? So let's just go down the track of humoral response. So in humoral response, the B cells, so first of all, the T cells, the helper cells needs to get activated, right? So T helper cells would bind with this antigen and would get activated, right? suppose first of all macrophage is gonna engulf this cancerous cell, right, or the antigen, right, and the T helper cells are gonna get activated. The B cells, the B cells would also engulf one of these cell that has the antigens, and the activated T cell would then bind with the B cell, and it would release this chemical cytokines, which would cause the which would cause the division of the B cells into B effector cells and B memory cells. The B effector cells would then differentiate into plasma cells, and the plasma cells are responsible for producing antibodies, right, and as memory cells are produced, these are going to provide immunological memory. So basically a vaccination kind of question. So where for the first time when the person is being exposed to the antigen, that is going to be primary immune response, um, and when the person gets exposed to the same type of cancer cell for the second time, that is going to be secondary immune response. So I have basically written all of that down. The viral envelope proteins will be engulfed by macrophages. The MHC of the macrophage will combine with the viral protein, and the MHC complexes will be represented on the cell surface membrane of the macrophage. The T helper cell will bind to specific antigen on APC, antigen representing cell, and becomes activated as well. B cell will engulf the envelope protein and act as an APC. Active T helper cell will bind to antigen on B cell and release cytokines, right? That would cause clonal selection of B cells. It would basically cause the B cells to divide and cause B cells to produce B effector cells and B memory cells. Right? B effector cell differentiates into plasma cells that produces up antibodies. When cancer cell will be produced, the person will undergo secondary immune response, right? Um, that would cause the antibody levels to increase rapidly. This will quickly destroy the cells by agglutination. It means it would cause the cells to stick together. Opsonation, it would act as a label for the macrophages, and neutralization. It would neutralize any of the toxins that has been produced by the bacterial cells. So yeah, we're done with it. If you do have any questions or if you are facing problems in biology and or chemistry, make sure to comment below. I will be more than glad to help you all. And yeah, if you find this video to be helpful, drop a like, comment, and subscribe. Thank you. Have a great day, everyone. Good luck.